If you follow us on social media, you'll know that we've been waiting for a big announcement. And today I'm talking to Alan Wolfe about a huge urinary microbiome study that will be the first of its kind. This groundbreaking work will be taking place in the US, but the research will have implications for researchers, clinicians, and patients worldwide. Joining me today is Alan Wolfe, Professor of Microbiology and Immunology at Loyola University Chicago Stritch School of Medicine, and co-founder and co-director of the Loyola Urinary Education and Research Collaborative, also known as LUEREC. Thanks so much for joining me today for our chat.
You're quite welcome. I'm looking forward to our conversation.
It's quite a big day for our respective teams because we're officially announcing our collaboration on a groundbreaking study into the urinary microbiome. But before we jump into discussing that, can you provide an overview of what is already known about the urobiome?
Sure. Maybe I could restate the question, which would be, how in the world did you ever get involved in, urobiome research? And the reason I say that is because I'm actually a microbial geneticist. My PhD is in genetics. I have a master's degree in systems engineering. And for the first 3 decades of my career, the work that I did was to simply try to figure out what the circuit board inside E. coli K12 looked like. So that's the laboratory strain of the bacterium E. coli. Um, I simply wanted to know how does a cell, a bacterial cell, uh, know how it's feeling? How does it sense its environment and respond appropriately? It's sort of one of those questions that falls under the category of, what is life, right? So, but about 16 or 17 years ago, I walked up to a world-renowned urogynecologist by the name of Linda Brubaker.
Mm-hmm.
I had no idea who she was. I had no idea how influential she was, but the chairperson of my department, Catherine Knight, told me that I should introduce myself. So I did. I walked up, I reached out my hand, and I said, "Hi, my name is Alan Wolfe, and we have two words in common: E. coli. You want to kill it, and I work on it." She said, "Let's talk." So we sat down for a series of 1 hour conversations in her office. And during one of those conversations, she said, some people think that the bladder is sterile. And I said, no flipping way. Have you seen plumbing? Of course, she's a plumber. That's what urogynecologists do. They fix the plumbing. As a microbiologist, I couldn't imagine how any part of the human body that has a hole to the outside could be sterile. I said, if the bladder is sterile, I want to patent it because something really special must be going on. Well, it turns out that it's not sterile, and I'll tell you how we know that in a moment. But something is special going on because, particularly in women, it's right next door to one of the most highly colonized niches, right? The vagina, and the difference in the amount of bacteria in the bladder and in the vagina is huge. Okay, so anyway, what Linda and I decided to do was to uh, withdraw urine directly from the bladder. This is when I learned about something called suprapubic aspirate, where you stick a needle in the lower abdomen into the bladder. Mind you, these were patients going in for urogynecological surgery. So the way I put it is, this is the least worst thing that was going to happen to them that day. They did, they weren't going to feel it. They were already out. But we took skin swabs and a sham needle stick, so a needle that went through the abdomen at the same place but didn't go into the bladder. We took urine by transurethral catheter, voided specimens, and vaginal swabs, and then we did something called 16S ribosomal RNA sequencing. There's a gene in all bacteria that is quite conserved. So it's very similar in all organisms. It's just different enough that you can tell one species of bacterium from another by sequencing the DNA in that gene. And so we did this, what's called 16S ribosomal RNA gene sequencing on urines, that had been, well, on these samples, and their urine had been sent to the Clin lab, and they had come back as no growth. So these were culture negative urines. And what we found was DNA evidence of bacteria in culture negative urines. We also found that the catheterized urine and the suprapubic aspirated urine looked very similar to each other. And since the aspirated, the route of aspiration bypasses vulvovaginal contamination, we knew that we were looking at bacteria that were in bladder urine, right? So the skin swab and the sham needle stick were controls, and the urine didn't look like those controls as well. And interestingly and importantly, the voided urine looked too much like the vaginal swab too often, right? So we published that work just about 11 years ago. But what the sequencing couldn't tell us is whether or not the bacteria that we had detected by sequencing were alive, because you can sequence DNA from dead bacteria. So the next step was to see if we could crow the bacteria. And another member of our team, Paul Schreckenberger, was the director of our clinical microbiology lab. And I sort of chided him. I kept bugging him and said, Paul, I'm looking at the taxa, the types of bacteria that we were sequencing. I said, these are not unculturable. They just don't grow under the standard urine culture conditions. Can you develop an approach that will allow us to grow these bacteria? So he invented what he called expanded quantitative urine culture, or EQUC. Rhymes with duck. It's an enhanced culture method. So it includes, we use a larger volume of urine, many different growth media, different atmospheric conditions. So not just ambient, what the air that we breathe, but anaerobic conditions where there's no oxygen at all, conditions where you put a little bit of carbon dioxide into a chamber. And that allowed us to grow a lot of the bacteria that we could sequence. And so now we knew that there were bacteria in the bladder. And the question was, did the bacteria in the bladder of individuals with different health statuses differ? So we looked at individuals with urgency urinary incontinence, stress urinary incontinence, interstitial cystitis, painful bladder syndrome, individuals with urinary tract infections, recurrent UTIs, people with kidney stones, and on and on and on. And what we found is that in many cases, but not all, that the communities of bacteria that we saw in individuals with lower urinary tract symptoms of some sort or another differed in important ways from individuals that were asymptomatic, presumably healthy, right? So now that we knew that there were bacteria in the bladder, that they were alive, and that they may in fact be associated with health and disorder, the question is who was good, who was bad, how do they interact with each other, and how do they interact with the host or hostess? In fact, most of the work that we've done has been with women. One of the nice things, scientifically very important thing, is that others had joined in the fray and started to report similar findings to ours. And we've grown as a community. We now have our own conference, which we started back in 2019, had another one in 2020, and then the pandemic hit. We're going to have our first Eurobiome meeting in March of 2023, where we can all get together again and try not to get COVID while we're there. To a large extent, and we and others are still looking at, how did the communities of bacteria, what we call the Eurobiome, differ between individuals with different health statuses or different demographics, different whatever you want, right? But we're moving, and how does the the microbiome of the bladder urine differ from the urethra, from the periurethra, from the vagina, from rectal swabs. I mean, we're really trying to understand the whole ecology of the
Mm-hmm.
system. That's where we're headed. We're headed towards ecology, right? You know, it's a niche. It's an ecological niche. The question is, you know, who should be there in a given niche? My niche is different than yours, so my urobiome should look a little bit different from yours. You probably have lactobacillus. As a male, I shouldn't have
Mm-hmm.
lactobacillus. I have no idea, because one of the things I haven't done is donated my own urine.
Oh, what?
I haven't done it, and we could do it. And I
It
just,
feels like something that you should do at some point.
it does, you know, but I just haven't. I don't know, maybe I just don't want to know what's
Yeah,
there.
I can understand that too.
Yeah. But in any case, uh, a lot of what we're doing in my group now is to try to understand who's good and who's bad. We
Mm-hmm.
know that some E. coli are pathogens. We know that organisms like Klebsiella and Proteus can be pathogenic.
Mm-hmm.
Same thing with Enterococcus faecalis. But there's a whole bunch of organisms that you may have heard of now, but most of um, your um, your community may not.
Mm-hmm.
Many organisms that are difficult to pronounce, and clinicians haven't even heard of these
Mm-hmm.
things. The question is, are these emerging uropathogens? Are these uropathogens that have always been there, but we're now beginning to realize that they're pathogenic? So
Right.
a big part of my group now is attempting to characterize the organisms that um, show up in case reports. Like those are
Mm-hmm.
one-off. Clinicians found some organism in an abscess or in a urinary tract infection or something,
Mm-hmm.
and they report it, but it's not science, it's anecdotal.
Yeah.
Well, so now what we're trying to do is ask, well, are those anecdotes, is there any basis to the truth of those anecdotes? Was
Mm-hmm.
that truly there because it was causing symptoms, or was it just an innocent bystander? And that's a non-trivial problem to solve,
Mm-hmm.
because no one's ever worked with an organism. You just don't know how to work with it. So
Right.
we're pretty much starting from scratch. And um, many of these organisms are difficult to grow. They don't want to behave.
Mm-hmm.
They're not model organisms that researchers have worked on for years. So that's where the community is right now,
Right.
and it's a great foundation for the study that we're about to launch.
So going back to that study, can you explain why it's so significant and why the bladder was not included in previous research published around projects like the Human Microbiome Project?
Right. So yeah, let's go back to the Human Microbiome Project. Now, I've heard this secondhand, okay? Not third hand, it is secondhand. There's only one person in between me and one of the members of the original HMP, the Human Microbiome Project.
Mm-hmm.
In fact, voided urine was included, but it wasn't included in the final manuscripts
Mm-hmm.
because the reviewers all claimed that what they had discovered must be contamination because the bladder was sterile
Right.
or urine was sterile. That's what I've been told, indirectly,
Mm-hmm.
and it rings true. Um, so right, the reason that people thought that urine was sterile and the bladder was sterile is it goes back actually about 160 years. Okay, if I can take you back in time, a little bit of history.
Mm-hmm.
For those who remember, in either high school or college, or maybe for those that were in really progressive school districts, right, in middle school, you remember back when you were learning about how there was this debate about spontaneous generation, right? Could um, life uh, come from inorganic, you know, um, could it spring up without having been born from something else, right?
Mm-hmm.
And after they figured out that maggots in meat were simply the larvae of flies, and if you
Mm-hmm.
covered the meat, right, it didn't get maggots. right? Then they went to smaller items, right? Smaller things. And at some point, they had completely narrowed it down to the only thing that could spontaneously generate would be these little microbes, the germs, right? So one of the key experiments was done by Louis Pasteur, who's essentially the founder of modern microbiology. And he did this experiment where he had flasks that he left open, liquid in flasks he left open to the air, and others that had a long gooseneck where, you could get air in there, but if anything was heavier than air, it would fall down and it couldn't get into the liquid. And he found that, all the various liquids that he put in the flask with the gooseneck, after sterilizing it, remained sterile, no growth. Okay. And if he left it open where things could fall out of the sky, it would turn cloudy, it would get turbid. One of the liquids that he tested was urine from someone who was presumably healthy. And the flask that was not really open to the air remained clear. And the flask that was open was cloudy. Now, what Pasteur said was that this disproved spontaneous generation. But about 15 years later, a very, very influential paper was written by a guy by the name of Roberts, who said that normal, healthy urine is sterile. That was his first sentence. I have no idea how he came to that conclusion.
Yeah.
Okay. But here's what I think happened. And that is, since we now know a couple of things. One, bacteria are very, very tiny. Okay. And you can have bacteria in urine up to over about 10 million bacteria per milliliter. And below that level, that liquid, whether it's urine or growth medium, right, will be crystal clear, right? Below 10 million, right? So that's 10 to the 7th colony forming units per mil, way above the threshold for the standard urine culture test, which is 10 to the 5, right? That's just 100,000 colony forming units per mil, or bacteria per mil, right? So first of all, a liquid with lots of bacteria in it can still look clear. The second thing is that most of the bacteria that we find in urine don't grow in ambient conditions. They don't grow in air. They like a little bit of CO2. They like not having any oxygen at all. And you need very specific growth media, right? And urine's a pretty poor growth medium. And so I think the initial conclusion, the interpretation, was a misinterpretation of the data.
Yeah.
And then you step forward to the 1950s and 60s when Ed Cass, a brilliant man who was an infectious disease doc at Harvard, at the hospital system associated with Harvard, was looking for a way to tell whether or not somebody had pyelonephritis. And he came up with what we're calling the standard urine culture test. And it worked great for pyelonephritis. But it was somewhere along the line, it was transferred to trying to diagnose cystitis. And I can tell you it doesn't work very well because no one's been able to agree on a threshold. So if you look in the literature, you have, clinicians and clinical microbiologists arguing back and forth over whether they should use 10 to the 5, 10 to the 4, 10 to the 2, 10 to the 3. I mean, and they're usually talking about E. coli. We now know that there's all these other organisms.
Yeah.
Right? So the reason that people thought that the bladder was sterile was the use of this test, the old history, right? When a clinician gets a report from a clinical microlab that says no growth, that sort of got translated into sterile. And then that gets taught to the medical students. But in fact, if you know how a clinical microlab works, no growth doesn't mean no growth. It means
Mm-hmm.
no growth of what they're looking for, which is known neuropathogens,
Right.
E. coli, Klebsiella, and so on. So I just think it's a comedy of errors. Also, a lack of thinking thinking outside the box,
Yeah.
which as a microbiologist coming in from the outside, I went, no, we have to reevaluate this.
Well, that's what we're about to do with this study, which is great. And it's
Yeah,
quite a big study. So we're looking to recruit 1,015 participants, which is huge. And in that number, we'll be looking at the urinary microbiome in people who do experience UTI as well as people who do not. Can you explain why it's so important that we have that comparison in this study?
right. Well, actually, we're going to be getting two samples from the people who have recurrent UTIs, one while they're having a symptomatic episode and one when they're not. And we're going to compare the community of bacteria, actually, the community of the community of microbes, so bacteria, viruses,
Mm-hmm.
fungi, everything that's in there, um, in the same woman when she's having symptoms and when she isn't, and see
Mm-hmm.
whether or not the communities differ, right? So you can imagine that, the reason that a woman might be having an episode, a symptomatic episode, is because for some whatever reason, uh, a pathogen that's part of the community blooms, and now
Mm-hmm.
there's a whole lot of them, and that's causing symptoms. Um, uh, the controls, the people who are unaffected by recurrent UTIs, will be used as well, I use the term control. In
Mm-hmm.
science, you need controls. I used that term when I told you about the very first study, right?
Mm-hmm.
We controlled for the suprapubic aspirate by having a skin swab and a sham needle stick. We
Mm-hmm.
controlled for um, the voided urine by having a vaginal swab, because the voided urine could be only urine from the urinary tract, but it could be contaminated with vaginal or vulvovaginal microbes, which in fact it often is, right? So in science, to do science, you need negative controls.
Mm-hmm.
You, you need them because you need a comparator, right? If you don't have a control, now what you have is anecdotal evidence, right? You don't have science.
Right.
Okay, so we need the controls to know whether there's any difference between individuals with recurrent UTIs, a history of recurrent UTIs, and women who don't. And we need the symptom versus non-symptom sample to determine whether or not there is any reason to believe that the microbiome changes and that change has something to do with symptoms.
Mm-hmm.
Now,
So,
whether those changes are a cause of the symptoms
right.
or a consequence of the symptoms is another story, which is something we won't be able to determine from this study. But the study should give us a lot of information as to where we should set our sights
Mm-hmm.
for future studies.
Yeah, it's all really exciting. So we'll have 300 controls in this study and 700 people that experience recurrent UTI. And we're also hoping to recruit 15 people that will send us a urine sample for every day for 40 days, which is quite a lot of urine samples. So
Yeah.
can you explain what we're hoping to learn from those people and why you chose 40 days?
Right, right. Um, to be honest, that to me is the most exciting part of the
Yeah,
study.
yeah.
So someone please volunteer, right?
Please.
Please. It Here's why. So I got to take you back in time a few years. I had a graduate student by the name of Travis Price. And Travis and I went to a conference in Seattle when he was just trying to decide. Well, he was transitioning from being a master's student in my lab to being a PhD student. And he was thinking about what his PhD dissertation project would be. And I remember we played hooky the last day of the conference, and we took the ferry across the harbor to some island. We literally just went across turned around, got in the line, and came back. It was just
Yeah.
nice to do. It was a cold day. I do remember that. And he was telling me about this crazy idea that he had. He was going to recruit a whole bunch of nursing students to give him samples every day so
Mm-hmm.
he could follow, you know, whether or not the microbiome changed, um, whether anything that they did in their lives, like, um, changed the microbiome, because he knew that there'd been a study like that done on the gut microbiome.
Mm-hmm.
By the way, it's just two people. One was senior, one was junior, and we figured that the first author was the student, and the second author was the mentor. And but it was a really fascinating, relatively early microbiome study where they sampled themselves basically every day
Mm-hmm.
for months. So in any case, he prepared. I said, okay, go for it, right? So he prepared for that by sampling himself every day,
Mm-hmm.
and a master's student, a female, sampled herself every day. And one of the things, two things that we thought that we, again, this is anecdotal because it's one-offs, right?
Yeah.
One of the things, besides learning how to do it and how not to do it, and finding out that it was feasible,
Mm-hmm.
what we what we found out from him is that uh, he finished his big comprehensive exams and took several weeks off and went to Thailand with his partner.
Mm-hmm.
And he thought, that's great. I'm going to be eating different food. I'm going to be in a different place. I'm going to come back. My urinary microbiome will be different. First day back, no different whatsoever. Right. Second day, very different.
Mm-hmm.
Third day, in the hospital with pneumonia.
Oh no.
So what we saw was that his urinary microbiome changed because he got pneumonia, and he could have known something was up the day
Mm-hmm.
before, except that we didn't get the results for like 3 or 4 days later.
Right.
His partner in crime was actually um, doing the analysis, and he was sampling himself every day, even though he was sick. We also got to see the antibiotics work, which was kind of cool.
Yeah, that is kind of
The
cool.
other thing we saw was that we saw, actually, I misspoke. They also did a little study with, I think, 7 or 8 young women for like
Mm-hmm.
13 days. We actually saw one woman get a UTI,
Oh yeah.
but the very last day, she walked into the clinic and said, I have UTI symptoms. But we could have told her a week earlier. We couldn't, because we weren't allowed to
Mm-hmm.
tell her anything, right? But we could see the E. coli working its way from, out the periurethra, which is the
Mm-hmm.
skin around the opening of the urethra, into her bladder, and the numbers growing. We have no idea what happened after that, right?
Yeah.
And the other thing that we learned from these small preliminary studies was that um, we think that binge drinking affects the urinary microbiome.
That is not that surprising.
No, no, it isn't. But here's what happened. So this was a master's student, and she had started sampling herself pretty much every day. In fact, at some point, she was sampling herself in the morning and the afternoon to see if different times of the day would
Mm-hmm.
matter. Um, and we got to the end of the semester, and what do graduate students do at the end of the semester? They
Drink.
celebrate.
Mm-hmm.
So she looked at her samples and went, "Whoa, something changed." And she was astute enough to think that, well, maybe it was because I had done a lot of drinking over the weekend. So we set up a small study to test that. And the committee that decides whether or not you can do a study on human beings is called the IRB.
Mm-hmm.
And we learned that the IRB won't let you say that you should drink on the weekends, but you can tell them not to drink during
Yep.
the week. So we set up a small study to see whether or not there was anything going on with binge drinking. And
Mm-hmm.
we don't have enough controls, we have enough data, but I anecdotally, I think that binge drinking probably does affect some of the microbes um, in in uh, in the bladder. But
Mm-hmm.
now Travis had the opportunity to set up a real well-designed study. And so he recruited 8 young women all in their 20s and 30s. All were students or employees of Loyola because we needed them to drop their samples off every day. In our buildings, they had to be able to have access. And they sampled themselves every day for 2 months, and then once a week for the third month. He asked them every day they had to fill out a questionnaire, which was incredibly invasive. They were asked about what they ate, any drugs that they had taken, prescription drugs or whatever, whether they bathed, whether they swam, whatever they did, right? And all sorts of invasive things about sex and contraception and so on and so forth. What he found was really quite striking. If we kept going back and forth when looking at the data, he would say, it's not stable at all. And I was saying, well, no, it is stable. He'd go, no, it's not stable. I said, yes, it is. Finally, I realized that, I don't know how many of your, audience members of this podcast remember Aesop's fables, Aesop's fables. The blind man and the elephant. Well, we were just tapping on the elephant from different directions, right? He was playing with the trunk and I was patting its side or its ears or something. So I eventually came up with a way of describing what we were seeing, which is if I knew what your microbiome looked like last week, and I know what it looks like this week, I can predict what it'll look like next week. Here's what I can't predict: what it'll look like tomorrow. Because what was going on is that there were essentially, in most cases, two different communities that were changing places with each other. So you'd have community A, and that would get replaced by community B, and then community A, B. And the duration of either A or B could be 2 days or 5 days or 3 days, and you just could never predict the next day. But you knew that in a couple of days, if you're in state A, you're going to be in state B shortly, with a few exceptions. And this is really intriguing because one of the exceptions, and it was without exception, menstruation caused a massive change in each individual's microbiome. So some women, there was one woman who was not on contraception. She was menstruating every month. There were some that were on contraception that where they went through the cycle every month, and there were some that were on this 3-month cycle contraception. The community, the change in the community structure was the same for each woman, right? So when a woman had menstruated each month, the change was essentially the same, but each woman's change was different, right? So there was a uniqueness. Now we only had 8 women, and so maybe there's like a handful of states that all women go into. Maybe there's 8 of them, 10 of them, 80 of them. I don't know, but so menstruation had this big effect. And the other thing that had a big effect was vaginal sex. And it didn't have anything to do with the male, because whether a male wore a condom or didn't wear a condom, there was always a disruption the day after, or almost always. And it was a particular type of bacterium that would show up, a bacterium called Streptococcus. And it would last for a day or 2 or 3, and then it would disappear, or at least diminish. So we think it has to do with the sort of the physical activity of vaginal sex, that the Streptococcus is innate to the young woman, and having sex was causing a disruption. And now the thing is that so what he did was to take he re-upped one of the woman who was having the most sex. So we had enough we had the most data points, and recruited her male partner. And
Mm-hmm.
they swabbed all sorts of parts of their bodies and gave us samples. And it was very clear that literally every time she had vaginal sex, streptococcus showed up, and it was a particular species of
Mm-hmm.
streptococcus that we managed to identify. And I thought initially that he was inoculating her with his
Mm-hmm.
streptococcus, but it turned out that this streptococcus was resident in her urogenital tract.
Mm-hmm.
Um, so, so here's the thing: I have a hypothesis that we've not been able to test,
Mm-hmm.
which is that maybe the urinary tract infections that come from that are associated, are often associated or thought to be associated with risky behaviors,
Mm-hmm.
may in fact simply be that when you're drinking and you have sex, you've got two things that are reducing the protective microbiome
Mm-hmm.
and allowing the blooming of non-protective bacteria. And if you're exposed to a pathogen like E. coli or Proteus, you're now more at risk. And I'd love to be able to test that, but it requires that I get money that I don't have to set up a rather large study. So back to our study.
Mm-hmm.
Why is this every day for 40 days important? Well, 40 days is we need enough data points to be able to tell whether we're seeing a pattern. The reason that we were able to detect a pattern in the original study was because we got something along the lines of 70 samples, right, from 8
Mm-hmm.
women. And so we were able to clearly see these patterns. And when there was something happened that changed the pattern, you could see it. If we only had a week, we wouldn't know whether we were seeing a random occurrence or whether it was actually indication of some pattern that we might be able to see if we had more samples. So
Mm-hmm.
40 days is really important. Well, the question is, are women with recurrent UTIs, are their microbiomes as stable and dynamic in the way that we saw for young women who
Mm-hmm.
were reasonably healthy, right? It, it could be that people with recurrent UTIs have a very, very different pattern, right? First of all, I expect to see different bacteria,
Mm-hmm.
but are we seeing, fluctuations A, B, A, B, A, B, or is it just like A the whole way and then all of a sudden you see B?
Mm-hmm.
They have symptoms, okay? So hopefully some of the people who are donating every day, I know this sounds bad, but get an episode so we can see, right? But it'll be random. I mean, hopefully for their sake, they don't get symptoms.
Exactly.
But from a scientific point of view, we'd like to see a couple, 2 or 3 of them get symptoms so we can use that information to design the next
Mm-hmm.
study.
Mm-hmm.
So yeah, the problem with most of these studies, um, two problems with the studies that have been done. One, they're way too small.
Mm-hmm.
Okay, so all the work that's been done with recurrent UTIs or UTIs in general have been on the smaller side. Okay, so you don't have a lot of power, statistical power. So your interpretation is well, we think we might have a pattern here.
Mm-hmm.
We might, you have to sort of be, if you're going to be rigorous scientifically, you need to sort of be a little cautious about your conclusions and your interpretations. But the other is that most of them are cross-sectional,
Mm-hmm.
and they're one time point. So you're looking at people that are presumably healthy, they have no symptoms, and people who have, I don't know, chronic kidney disease.
Mm-hmm.
You look at them and you go, well, this group looks different from that group. That's all you can say.
Yeah.
But if you were getting samples every day from these individuals, then you could determine, well, how did they become? I mean, for example, I'd love to be able to get somebody who's going to get a UTI, okay, and start sampling them,
Mm-hmm.
a month before they get the UTI. Let's say we could predict you're going to get a UTI on March 1st. I'd say, okay, great. Let's start sampling you every day starting on February 1st. You're going to get a UTI on March 1st. You're going to get treated, and we're going to watch the treatment work or not work. And then
Yeah.
we're going to wait, after the treatment, we're going to wait another month to see if anything changes. Okay. So,
Mm-hmm.
that's ideal, because then you can really tell what's happening. And from that information, you can then begin to determine, well, is there anything, do we understand the process? If we understand the process, then maybe we can intervene, right? Maybe at least we can predict, right?
Mm-hmm.
Like, wouldn't it be great if you could take a test every morning, when you're, before, after you brush your teeth or whatever, and you look and go, okay, today, tomorrow, the day after, I'm gonna be fine. But you look down and you go, I've got a biomarker. I have a marker here, right?
Mm-hmm.
I'm going to be, I'm going to have a UTI tomorrow, and perhaps you could do something about it.
Yeah,
That
that
would
would
be
be
great, wouldn't
ideal.
it? I would love
Very.
that, right? But I mean, we can't. We don't know what those biomarkers are. We don't even know whether you can tell the day before, although we suspect that you can, based
Mm-hmm.
upon that anecdotal data that I told you about.
Yeah.
So the everyday sampling is really, really,
Mm-hmm.
really, really critical for us to get a first look at what a daily microbiome looks like in women that have a history of recurrent UTIs.
Yeah, it would be very interesting for us. And alongside the urine samples, our team has developed some very in depth surveys which mimic a little bit what you explained earlier, where we'll be collecting information about all sorts of things that could impact the urinary microbiome. So, what
Right.
kind of associations are you anticipating that we might see between the survey data and the urine sample data?
Um, age,
Mm-hmm.
uh, clearly, um, recurrent UTI versus controls, um, symptom versus asymptomatic,
Mm-hmm.
episodes. Um, we maybe location, geography. I mean, I don't know. I mean, most of the work that's been done has been on women from either Chicago or Portland, San Diego, a little bit from Providence, some work in North Carolina, a few other places, some work that's been done in China, a little bit of work that's done in um, certain parts of Europe. I would presume that that women in North America might be generally similar,
Mm-hmm.
but who the heck knows,
We'll find out hopefully.
right? We'll know whether um, certain medications that are allowed to be included in the
Mm-hmm.
study have an effect. Maybe if we get a large enough sample size of individuals on, medication A versus B
Mm-hmm.
versus none at all. Um, Geez, almost anything. I
Yeah.
mean,
Yeah.
the point is, no one's ever done this.
Mm-hmm.
So predicting the patterns outside of the obvious
Mm-hmm.
uh, is almost impossible. So, like I said, the obvious is I fully expect that symptomatic episodes and non-symptomatic episodes versus not, and women with UTIs versus the controls, I expect them to be different. I expect that on average, young women with recurrent UTIs or controls with that are younger will
Mm-hmm.
look different from the women that are older, particularly premenopausal versus
Mm-hmm.
postmenopausal. We already know that that's true, but this will be the biggest study where we can simply ask that question.
Mm-hmm.
Okay. But outside of, like I said, the obvious things, it's hard to know. But I know I've seen your questionnaire, or at least I've seen a draft of it, and it asks a lot of
Yeah,
questions.
we're quite excited to see what happens.
I think that the vast majority of questions, will elicit no pattern whatsoever. But
Hmm.
I guarantee you there'll be some that we'll see patterns, and I think that I'm expecting at least one or two surprises.
I hope so. And
Yeah.
with the information that we'll be collecting, what information can participants expect to receive after we finish with the data collection and analysis?
Well, you and I have been talking about this, right? I
Hmm.
mean, we're not there yet, so we're not exactly sure. Um, but one thing that we've tossed around is the idea of sort of regular updates,
Hmm.
you know, um, where we can talk about, well, how many individuals have we recruited and how many of those samples have been sequenced and sort of generally what we're seeing, not specifically, maybe. And then at the end, though, there will be a sort of a very de-identified summary report,
Hmm.
right, where participants could take a look at um, sort of what the generally what we found from the entire study. And then we will ultimately take all of our data, analyze it, process it, and turn it into manuscripts.
Hmm.
So individuals certainly, not just participants, but everybody who has can get access. And we should be able to put our publications up on your website.
Hmm.
Um, whether we do it open source so that we can put it up immediately, we'll have to pay for that, right? Or after I don't know, a year's embargo, but those manuscripts will be available. And our task will be to try to write these papers so the general public can understand what the heck we're talking about.
Yeah.
I try very hard to write in simple English, even when I'm writing to other scientists, but it's not easy. So yeah, it's not like the participants samples are going to go into the ether and or in through the mail, and you're never going to see what came of it.
Yeah.
We plan to make this available to the general public.
Yeah, it's definitely a goal of ours too to share the information as much as we can. And it's so important to our patient community to see what comes out of this study. And so the study itself will be, because of logistics, will be recruiting from US mainland only, but the findings of the research are likely to have implications globally. So how do you think this study might benefit researchers, clinicians, and patients in the future?
It's information. Um, it like I said, it's the biggest study to date.
Mm-hmm.
It is big enough that it will have statistical power for a number of different hypotheses that that we will propose.
Mm-hmm.
Uh, and since we will be making the information available to everybody ultimately, it means at the very least that clinicians who take the opportunity to read
Mm-hmm.
may open their minds, make them think differently. For researchers, it'll be a wealth of data to develop their own hypotheses and design their own studies. The goal, of course, is to get where we're at to where we need to be able to propose um, more accurate hypotheses for the underlying mechanisms
Mm-hmm.
that cause symptoms of things like recurrent UTI or urgency urinary incontinence, all sorts of lower urinary tract symptoms that, millions of people, particularly women, suffer from. Um, the if you understand the process, then you can begin to figure out ways to diagnose more properly,
Mm-hmm.
right, more accurately. Um, we know that the standard urine culture test, well, um, it is what it sounds like. And there are better diagnostics. The problem is that, like I said, we don't really know who's good and who's bad
Mm-hmm.
outside of a few that
Yeah.
have always been thought to be, pathogens. We're working on that. We have some pretty good ideas, but we need better diagnostics that can give an accurate assessment of what the ecology of an individual's bladder or entire urinary tract looks like. Then with some algorithm, treatment algorithm that takes into account not just the bad guy,
Mm-hmm.
right, the E. coli, but the rest of the ecology. So, for example, and it's an analogy that I use. Suppose you didn't know what the concept of a lion was. You'd never seen a lion. You had no idea what a lion was. And your friend said, "Oh, you want to see a lion? Well, there's a lion in a zoo down the road there. It's a pretty lousy zoo. The lion's in a cage on cement, and every once in a while they throw some raw meat at it. That's a
Mm-hmm.
lion." Or your friend could say, "Hey, you should book a ticket to Nairobi or somewhere else, in the appropriate parts of Africa, right? And go on a safari and go out into the savannah, and you'll realize that lions actually, well, they're males and females. They live in prides, families named prides. They spend most of their day lying in the shade under trees.
Mm-hmm.
Every once in a while, the whole group gets up and chases down a wildebeest or a gazelle. And the gazelle or the wildebeest is there because there's a water hole and the savanna is grassland, right? You're looking at the ecology. But, we're still living in the 19th century where it was important for clinicians to be able to say, "Oh, that's cholera. I can identify cholera and I can kill it."
Mm-hmm.
Okay. Or, that's salmonella, right? Or that's tuberculosis. That's the bug that causes tuberculosis. Those are truly invasive, true pathogens. But in most cases, what we're dealing with now are not true pathogens, not true invaders. They're not like COVID, which is
Mm-hmm.
clearly not something that normally lives within us. It's an invader from the outside. That's a true pathogen. We're talking about organisms that normally we live with, and normally they're kept under control, but something happened. You get some sort of dysbiosis, a disruption of your ecology, and now you get symptoms. Okay, so you need to be able to understand what the ecology is supposed to look like. And that includes not just the potential pathogen, it includes the rest of the urinary
Mm-hmm.
microbiome, and it includes the host or the hostess, right? I mean, right? The savanna, right? And my hope is that scientists, clinicians, clinical microbiologists, that everybody begins to start thinking about, well, let's think about urinary tract infections and other sorts of more chronic diseases, not just in the urinary tract, as disruptions or dysbioses
Mm-hmm.
of the niche where the symptoms are occurring. And then having treatment
Mm-hmm.
algorithms that allow us to try to restore what would be a healthy microbiome, or would be considered to be a healthy microbiome for that individual. Now, that's a pipe dream.
Mm-hmm.
I'm fantasizing here, but I think that ultimately we can get there. But we can't get there unless we do studies like the one we're about to embark on. We need the data. So we need people to volunteer to participate.
I
Okay?
hope they will too, and we'll be relying on our community a lot for recruitment for this study, so I hope anyone watching this video shares it. I also look forward to chatting to you more throughout the study so we can share updates and hopefully do some live events so people can ask you questions about how it's going.
Sure, why not?
Well, thanks so much for joining me today. It was great to chat.
Absolutely. It's always a pleasure.
Anyone interested in participating in the study can find the link to the screening questionnaire in the description below. The screening questionnaire will allow potential participants to tell us a little bit about themselves so we can allocate them to the right group if they meet the inclusion criteria. We're really excited about this research and we'll need the support of our entire community to recruit the participants we need. So please do share this video, subscribe to our channel, and don't forget to tick the bell.